Welcome, everyone. It's my pleasure to introduce a Professor Amanda Myers, Associate Professor Amanda Myers, Dr. Amanda Myers, to the a metadata scientist regular meeting. As you know, this series of talks are intended to introduce the broader scope that data science has these days, right? The many ways in which the application of data and computational methods to science is spreading across many disciplines. So Dr. Amanda Myers, as I said, is an associate professor here at the university, and she has been studying neuroscience since 1993 and human neurogenetics since 1995. She received her PhD from Washington University in St. Louis in May of 2002. Title of the dissertation is The Search for Novel Loci Involved in Late-Onset Alzheimer's Disease. And while at Washington University, she won a John Merle Fellowship, an Alzheimer's Disease Director's Education Award, and was a finalist for the O'Leary Prize for Outstanding Research in Neuroscience. Amanda performed her postdoctoral fellowship in the Laboratory of Neurogenetics at the National Institute of Health under the supervision of Dr. John Hardy, and was funded by the National Academy of Sciences during that time. She was nominated for a Distinguished Mentor Award during her postdoctoral work. Dr. Myers has received support from the National Institute on Aging, the National Institute of Mental Health, the National Institute of Neurologic Disorders and Stroke, and the state of Florida. And her current research involves looking at transcript and protein expression in the cortex and how that is potentially controlled by DNA variants, both in normal and Alzheimer's disease brains. Thank you so much for being here, Amanda.
Oh, thank you for inviting me. I really appreciate, leadership and the invite, and I really appreciate the opportunity to speak today. I think, I've been working with the IDSC or adjacent to the IDSC since it was called CCS. And I think it really makes a unique and outstanding place. So I'm really glad the IDSC is here. It's maintained its autonomy, it's cross campus. I think it's a really great thing. It's one of the things that works. Oh, and I should say, let's wait for questions in the end, but you are free to fill up the chat box with questions and I can just go through at the end, but sort of storytelling here. So I think it's better to just go. So Nick said, if I can get these slides to go, Nick said that I'm supposed to talk about myself, which I'm not great at, but since this is a meet and greet, I think I framed the problem as sort of, how did I get here and what made me keep in academia versus a lot of my incredibly smart friends who left. So traits that got me to this state will be the first part. And then I will be talking about data, but with the caveat that I've sort of geared this towards a 10,000 foot view with students. You can go to my webpage. Sorry, this link is crazy. I'm working on that, but you could just Google these letters, LFUN, and my name, and it should populate. So again, it's going to be storytelling, less hard math. I hope you can hang in. So who is me, the scientist? One person's experience. Again, my opinions are my own, but I do stand by them. So, your mileage may vary. But I think it's interesting. I've realized that maybe there is a question who is a data scientist. I do describe myself as a data scientist, even though my PhD is in neuroscience and my training is sort of genetics based. And that is because I generate large data sets. I look at large data sets, analyze large data sets. I think deeply about large multimodal data all the time. And I think for me, irrespective of how you formally define a data scientist, I'm a data scientist. I think why there could be questions about that. Again, I've never had formal training in even genetics. I've never had formal training. And I think this, speaks to my career that I'm still here. I really am a boots on the ground kind of person and learn by doing and pretty aggressive about, okay, I don't know how to do that. Let me go get information. I don't have any sort of pride. I do think that this creates holes in a career, but I think why I've persisted is I am deeply, deeply, deeply curious. So I'm absolutely insatiable. I think everybody has a ton of ideas, but I have ideas that won't let my brain go. They knock at the door of my mind and I just get obsessed. And so I think if you have that, gut in your belly obsession about your ideas and getting your ideas out there and executing your ideas, and that is, it's like candy to a baby, it's the most important thing for you, then I think, academia is a really good fit for people like this. The other thing that was really helpful is mom and dad. Hello, mother, hello, father. These two people met at Northwestern as they were getting PhDs. Everybody in my family has PhDs except for my brother. Don't feel bad for him. He works for Oracle and he makes a lot of money and is doing great. So, we're all sort of scientists elsewise, or academics formally. So, you look at this picture, this is mom, I think in the 50s in a lab. Of course, Amanda's a scientist because her mother's a scientist. But my dad was actually just as informative. So my dad was in business, ended up rising up in administration in the Haas Business School to sort of upper echelons there. This is him teaching a bunch of CEOs back in the day. He started out teaching and sort of, moved along back when people wore suits to class. But he is studying business as sort of a cognitive construct. Like, why do people buy? And I think through either brain wiring from the genetics or through talking to him, that really got me into neuroscience, very interested in how people think, why they think, what goes wrong with brains. So that was as informative as, of course, my mom being a scientist, incredibly helpful. And mainly because she gave me the gift of just playing around in a lab very early on. She had to create labs for the pre-meds for organic chemistry. And so she did that by making sure that I could do the labs so that they wouldn't be too hard or inappropriate. And so I've been in a lab for a very long time in my life. That really helped me. I don't think you have to be born from academics. I think that's pretty rare, but I would say trying to seek some real experience in what you're going to do, getting trained and becoming and doing what an academic job is so different. And so, trying to figure out what you're going to be doing at the end of the day, I think is important. And it can give you confidence and, understanding what information is absolutely power here. And so understanding what process is and the crazy aspects of getting funding and all sorts of other things is crucial. The other thing that has really helped me, and what I've been retrospectively in my old age looking back at, is everything is people. Universities are people, funding institutes are people, it's all people. So these are the people that have helped me, advisors, people that weren't my advisors, but basically helped me get my PhD, people that sat on my thesis committee that were, very fancy now, people that are friends that I met that stayed in science, friends that have left science but are just as informative to myself, technicians, all the people that do the work with you are so crucial. People that I've trained and are now doctors, they still help me. I ended up teaching medical students, so having that experience was helpful. People that can get funding very easily, those are helpful. Collaborators, and then, friends that you've met along the way and are really helpful to be at your level. I end with this guy. He looks very scruffy, but he's pretty fancy. His name is John Hardy, and he's a Breakthrough Prize winner. That is the US's equivalent of a Nobel laureate. It was started by, Facebook, Google, all of those guys. So it's a Nobel laureate and with a lot of prize money along with it. I'm ending with him because my career wouldn't be my career without my ideas and myself, but also without him. And the crucial aspect is that John and my personalities were very well matched. And I think that is a crucial thing to think about when you're going for different career stages, is to make sure you get with people that are a well matched for you. So I almost didn't have a postdoc because the guy that I was supposed to work for didn't come. I asked John if I could work anyways. And John's, thing to me was, I'm high up in the field. I don't want to teach anybody. I don't want to be a mentor. If you are coming here, you will receive no hand-holding. If you're willing to accept those terms, go for it. Now, I think a lot of people would have said no because they want to have mentoring. But for me, micromanagement is death. And so I think that's because I was, puttering around with mom in lab for a very long time. I am really more happy figuring out things on my own and then coming to ask for bits of help when I get a block, but really trying to, self-direct. That worked out really well. The other thing that worked out really well is John, part of the reason why he's a Breakthrough Prize winner is he is very much a person that gets very bored very easily. So he is always going for the new. So the fact that we were at NIH, that, things are a little looser there because you're not constantly applying for money and making sure that all the money is right. There's a little bit of a space. So I had the idea that I had in grad school. It was sort of shut down for various reasons. I then, brought it to John and it launched off. And it launched off because John is very, very open to new ideas. That is what thrills him. And it also was the space that we were in had some protective space to sort of test things. And there wasn't such a, everything has to work, you can't have a failure. So, people are really why I'm here, as well as my background experience. And that's just as much as grades or programs or whatever, it's people. So I would really argue to cultivate your scientific relationships and try to get in with people that are really a good personality fit for you. If you like a lot of mentoring, get somebody that's really going to do a lot of mentoring. If you hate sort of, all of that, just get with somebody that's going to fit with you. The other thing that I'm appreciating, that's not really why I'm here, it's almost why I'm not here, is as I go up, and maybe this is because fields are changing, but there's a lot of toxicity and a lot of people treating people really badly. I think the best way to deal with that is to flee. That's what I do, but I've gotten stuck. And so you can't always flee. You do have to, be beholden to the project, get your papers, get things done. But when I'm stuck, to keep myself sane as I yell and scream to my friends, but I also think about like, okay, how would you solve this problem if you were studying it as a scientist? So things like game theory, game theory is basically looking at this problem, which is called the prisoner's dilemma. And it's a social problem of how you win at the prisoner's dilemma. And game theory provides some solutions to that. So, thinking about things more abstractly was helpful for me. The other thing that's super helpful is the non-academics in your life. So whether that be your kitty that greets you at the door at night, or it could be your family that's not in academia that's really supportive of you, or your kids, that all works, but somebody who's not so imbued and invested in this space. These are my non-academics. They of course care about me, and so they're happy when I get good papers or whatever, but it doesn't, the valency is just taken out because it's, they're not in the space. And so it really helps you to keep perspective. I am wittering on about this because this was one of my best friends from grad school. His name is Scott Smemo. Met him in grad school. He was a great scientist. He got in a very, very hyper competitive lab in his postdoc, one of those dog eat dog death situations, step on your mother. And his brain is wired such that was a really bad situation for him. And he ended up killing himself. And he is not the only academic that I've known that's gone down this path. So again, look for your pressure release valves, because this is not where you want to be, and there's no reason that you have to be there. Academic science is not worth it. It is lots and lots of pressure. It's fun, it's great, fantastic, but you need to think about ways to survive mentally. So enough about the whole history. Let's talk science. Again, I don't know if there's students here or not, but I've geared this towards students, so it's going to be 10,000 foot view kind of stuff. I listed a lot of funders. Mainly right now I'm funded by the National Institute on Aging, but I would like to highlight that the IDSC, I hope they still exist, have these collaborative data science awards that are really great. It's tiny bits of money that you can apply for that really help to get stakeholders in projects and say, you're starting a collaboration. Oh, we've got a little bit of money that really, keeps the ball rolling. So this is really great. What I do in my career and where that came from. I think it's a really interesting thought problem to ask people where their ideas came from. If they are the ideas generators, they will have a tendency to witter on and on about where their ideas came from. So there always is a backstory. And a lot of the times in terms of a generation of ideas, at least for me personally, it comes from failure or stress or something where I think I'm going to fail. And so I had a torturous PhD where I had to sort of stop halfway in between because my bones were fusing together and I had to have major surgery. So it was, academics in or out. I took a year off. Am I going to do this or not? And I really did a lot of soul searching and really decided I don't know how else to be other than in academic science and at a university. So I fought to get back. And when I came back, rightly so, my advisor was very questioning whether I should stay in the lab or not, and basically saying, I don't think you should get a PhD. So this is another thing where, the business sense comes into it. When you have something like that, when you get into a negotiation space, know your red lines, know what you will not give up on. And I knew from that year of soul searching, I was not going to give up on getting a PhD, but also always offer up something. And so what I offered up was I would dump 4 to 5 years of work. I had this other project that I was doing, no papers, no nothing, just throw it in the trash. And I would move to this project, which I could do from my bed if I was sick. And so it ended up going well. We got a science paper because I was this had to work. And so I really, really did a lot of due diligence to make it work. The problem with it at the end of the day, it worked. We've got a spot on chromosome 10 that's involved in Alzheimer's disease. Fantastic. But, I was putting myself under pressure to find the gene, and other jerky people were thinking as a junior scientist that this was my job to find the gene. So we did a lot of work to try to find this gene. And taking a step back, I realized we were never going to make it. So we were looking at DNA only in a set of pedigrees, and the concept is called linkage. But if you don't have lots and lots of generations when you're doing linkage, you're stuck. You're not going to, doing all sorts of stuff with DNA is not going to answer the question. So this is, sort of the genesis of the idea out of failure. So I was thinking, grew up in Silicon Valley, data, computers, all of that stuff, everything is a data problem. So my idea was, what data are we missing, and how can we include more information to solve this conundrum of, we've got a big region of chromosome 10, but no genes. And so the idea simply that I proposed in my PhD was to get RNA on these families. And if you believe in Watson and Crick, DNA makes RNA, makes protein, and that's the only way that DNA can act on disease. So this is what we've mapped, is there is DNA somewhere on chromosome 10 acting on disease. It has to go through RNA. So that sort of got shut down, but then I went and met John, who was very excited. And it sounds such a simple thing, but I'm telling you at this time, this is not how geneticists thought at all. People were really, really focused on DNA. So how did we get this done, and how did I work with John? So mainly, the other great thing of John is that he not only likes the idea, he really supported me in the idea. He really threw me at, oh, I don't have time to go give that talk, Amanda, go do that for me. And so that is stressful, but it's also really great as a junior scientist to get as much experience as possible. So we needed human brain tissues. If you're looking at expression, you want the tissue of interest. So again, this is prior to everybody doing this kind of research. Nobody really thought about this, and we need a lot of human brain tissue. So we had to sell. So again, Dad's in business. One of my jobs as a kid was to run focus groups, is the art of selling. So how are you selling yourself in science? Like, why should you invite me to this lecture? As well as, how are you selling your project? And so we had to really tap dance to get people to get on board, because science in terms of Alzheimer's disease in the US is funded by these centers. They all have brain banks. Those brain banks were ignored. The pathologists were really thinking, one at a time, like, okay, there's a weird case, I'm going to take a bit of tissue, stain that, and look for pathology. But they weren't thinking in this so many different samples and looking at connections between DNA and RNA. So it was a reframing of how they acted, and they were sort of grumpy because they had been ignored. So we needed the heads of the NIA, of the Division of Neuroscience, to get on board. So gave a little speech, got them on board. They ran around and said, this is a good idea. Once you get your stakeholders, things go much better. So it ends up being a worldwide series, because we got the US samples, and then we got papers, and so everybody got invested. So it was easier to get samples from Europe. I do think it's a good idea to try to, in projects, go outside the US. Europe exists, the UK exists. It's still there, even though Putin's invading. But this really helps the series. The other thing that helps is there's 2 different series. It's not a US, Europe, across the pond divide. It is really 2 different collections that define the series, but half of it is Alzheimer's disease. I study Alzheimer's disease not because of great love for Alzheimer's disease, but because I have a love for genetics and neuroscience. Alzheimer's disease has got some great things about it if you want to study genetics and neuroscience, namely that there's a lot of people that develop Alzheimer's disease, so it's common, it's very genetically driven, And then the other thing is it's got this really defined pathology. And having defined pathology is very useful when you're doing things like dialing through all this data, because you really, ideally you want to test any finding to make sure that it's really relatable to your disease pathways. And so having defined pathology is helpful. It's a lot of brains. I run only a portion of them. And I do that again because of potential for failure. This paper, I had to make it work to get my PhD. There was no going to another project. And so I was very careful about who got into this paper and who didn't. Somebody went and decided to not use Amanda's special sauce selection and reran everything. The peak's still there, but it wouldn't have been a science paper. And so, denoising the data and really making good a priori decisions about who to run and who not to run really helped me. And so I do run the cleanest portion of my data as possible. And it's DNA, RNA, and protein. It is nothing fancy. And preps, people do fancy things. I won't dial into the reasons why we don't. I don't know if people came on early. I do have this video that sort of shows the concept of our old pipe. I won't show it because it's the old pipe. It's on the webpage. The old pipeline results in sort of two pools of data. Networks, which I will be talking about because we're really focused on that for the new pipeline. But also, I'm going to touch on this a little bit because this is what I'm funded for through the National Institute on Aging. So it's networks. That is what people think of when they think of this large data analysis, big sets, looking at all the data, making connections. You can also sort of do things one at a time. So, very simply, you can look at how DNA is controlling each single RNA target over and over and over again. And that you can visualize as a series of box plots, whereby you are looking at with each change, with each additional copy of a potential risk allele, this is real data, you are changing expression. And so, you have controls in black, cases in red. And so, for example, here, you've got two copies of a risk allele that is driving expression. It's driving expression in controls, but in the cases, it's driving it even more. So this is really interesting. You've got this difference here. So what NIA has funded me for is to look at that difference. We are capturing that by going back, doing other types of sequencing, and really reprofiling data. But what the IDSC funded me for is to look at the networks. So there are many, many, many different ways to build networks. It's too much to go through all of them, but we have made a choice for very specific reasons. And what we do is something called Bayes. And these are the reasons why. I'm very into what this means, mixed data types, means that I'm interested in looking at DNA and RNA together. So all of the RNAs and all of the DNAs as much as possible together in the same space. That ends up being a mixed data types problem because DNA is a 0-1 signal, RNA is a continuous signal. So you need to treat this differently to get this to work, and Bayesian networks really allow you to do that. The other thing that I'm really interested in is causality. One of the things that people were really criticizing when we first started this is postmortem tissue, end of life, people are dead, you're not following up every single process that's changing throughout time with that person. Are you really getting anything of interest? And so, are all of the effects that you're mapping just reactive? And, how you get around that is, for me, you incorporate things like DNA, but you also do the appropriate modeling to get at as close as you can computationally to this causality, which would be, things that are really upstream of disease as opposed to downstream of disease. And so that's another reason why Bayes is great. The other thing that I really like, I'm calling it feature classification. I don't know if that's the right term, but there's a lot of networks that don't have any sort of bookkeeping. So when you're doing Bayesian and when you're building your connections, you're keeping track of all the targets. If I was studying a problem like I want to find pictures of my dog Mitzi, I would definitely not do Bayesian because it is very slow and torturous. You can do much better predictors through other ways if you're doing like just a feature classification problem. And so with feature classification, AI kind of things, deep learning, you don't really keep track of who is in that predictive model because you don't care. All you want to do is find pictures of your dog Mitzi. I do care. I want to both predict Alzheimer's disease, but I want to dial back into those targets and understand who they are and why they're doing what they're doing. So that is why we do old school, low and slow Bayes. What is Bayes? A lot of people in the room probably know this, but it is just simply doing a data search, including all of the data that you already know from prior search. So it's like, trying to find something, walking around with the backpack of all the information you've already got. And so prior data informs prediction. And so this is where hopefully everybody is still awake and you can type in chat. The classic example that I have stolen from 3 Blue 1 Brown is people with these traits. If you are in Main Street average, everyday America, if you ran into a person like this, would they be more likely to be a farmer or a librarian? Please type in chat. Anybody? Come on, Alberto at least. Okay. Farmer, oh, because, okay. Librarian, not so easy. Tim, yes, not so easy. Farmer, librarian. Oh, it's sort of even split. That's quite interesting. Maybe people know this classical problem. So what the answer is, and the key bit is what I said was Main Street America, is that proportionally speaking, in America as a whole, there are just more farmers out there. And so both of these traits describe both farmers and librarians. And if you were doing this problem and just limited your sample to the university, of course, you're going to find more librarians. But the prior is the distribution of the numbers of people. So if you have more farmers, you are more likely to find a person with these traits to be a farmer, just proportionally speaking. So that's why Bayes is really powerful. Bayes is used for search to find, treasure, to find down planes. Somebody probably has written it to, find your keys. It can search and find everything. Its problem is that it's slow because it's keeping track of all the relationships. But it is good to keep track of all the relationships because then you have biological interpretability. So how we do this in our data, I like to visualize this as a data battle because that's fun for me. So you are basically searching through your battlefield of all of the data and scoring relationships. And this gets at something called conditional probability tables. So to visualize this, you can visualize a 3-way pair. And the first thing that you map is, okay, these 2 targets, let's say it's RNA, those things are correlated in a linear fashion. Fantastic. That's just co-expression. That's just saying things are going together. But what you also get is other information. So this is your prior. You are looking at, okay, so it looks like DNA is also correlated with this Y1 target. So that's great. That's your prior. I want to fit that into this model, into this relationship. How does knowing about this correlation inform this correlation? And if what you get at the end of the day looks like garbage, that is actually information, because it means that this relationship is really explaining a lot of things about this relationship. And so because of this effect, that helps you order the data. So you do know that there's a relationship between Y1 and Y2 because it's correlated, but the primo relationship is that's really driving the effect, and that's because this breakdown is between X and Y. And so that's how you order things. And it's way more complicated than just 3-way. Again, it's a whole huge battlefield of data that you are testing and testing and testing and keeping track of all of these orders. And so by iteratively testing, you can really, organize the data. The formal pipeline is here. I'm only really talking about a few main aspects of it. But again, we analyze 2 data sets and run a whole bunch of different tools. Again, with the real goal of getting biological interpretability. I'm very focused on not only the good math, but what the good math is giving me. So finally, let's get to some outputs. What have you found? These are the really awesome people. I really love this collaboration. So it's people at the City of Hope. Andre is the guy that chairs the entire department and the original sort of hook for me. Sergio is the guy that really helps to think about problems and run a lot of the code. And then when we break everything down, Grigori is this key. You really need to have programmers involved so that they can help you fix things as you're breaking things. So all of these guys and myself really are team science here. So we had two goals. Process goal, again, focused on mixed data types. That's really crucial to answer the question that I want to answer, which is how is DNA controlling downstream expression of anything? John Hardy, again, is informative here because he always said to me, keep it simple, stupid. And keeping it simple would be to, again, I'm asking how is DNA controlling things, want to get DNA in my model. We picked ApoE because this is a target that is so robust in late onset Alzheimer's disease, it's very hard to kill. So if we didn't find this in developing these new models, then something was wrong. So fortunately we did. This is just the runs. What this means is set 1 was run subly from set 2, so different battlefields. But again, everybody is equal in this battlefield. We have not rooted ApoE in the model. We've not given it armor. We've not declared it general. It just is another foot soldier fighting it out to try to win and be informative. So it was really nice, first of all, that we found ApoE in both sets. It is much stronger. So what this number means is that knowing whether somebody is ApoE positive or negative can tell you a lot whether somebody has disease. And so each of these things on their own, knowing how VGF is can tell you whether somebody has Alzheimer's disease or not. Things downstream are connected, particularly structures like this. You can know something about diagnosis from knowing the state of this, but we have to go through other targets. So these top bits are sort of our key targets because they're really independent of each other and they have strong weights to prediction. So you can see here in set 1, we've got a really strong effect with ApoE, less of an effect with ApoE in set 2, and just less data in set 2. So the sort of one of these another fail states, you're in a good collaboration if when you fail, everybody rolls up their shirt sleeves and gets to work. Why aren't we reproducing? Because I had another paper with this old pipeline where we got really good reproducibility. So how to address this problem? If you think about this data, again, there's two things. It's a mixed data type. We're looking at expression, and then we're also now bringing in DNA alleles. So let's, we decided to formally mathematically look at this. I will skim over this, but this is, really what was done. It's a statistic called the Jaccard Similarity, which is a very simple statistic, but basically we calculated this comparing our two data sets, the real data, which is in the blue line. And then Sergio also, predicted based on a particular distribution, random data, and then, mapped that. And so the idea here was, is the similarity predicted between our two data sets higher than what would be expected by random chance? And so it is that space here, it means that we've got better similarity in our data than random chance. And this is the p-values behind that. So this was great. This really indicates that I don't have to retract a paper because the data sets in terms of thinking about expression are very similar. I'm sure you can think of where they go wrong. It's in the DNA. So set 1 has a lot of people with this risk allele, set 2 really doesn't. You can see that from the structures, we just have a less of an effect of ApoE in this set 2. And that really does make for a very different structure because it's power, it's the number of people that are contributing to your effect are less. And it's also, anytime you include anybody on that battlefield, if they're an important part of the battlefield, they are going to change a structure. So you really do have to have very comparable data sets when you're trying to go for reproducibility. And so this was a problem. So we decided, okay, let's deal with the most powerful data set and really dial into data set 1. But you always have to think about rigor and reproducibility every time you're doing science, that if you are a PI in academic science, that is make or break your grants. You have to do this, thread this loop of, I want you to do something really novel, but I want it to work. So it's always thinking about how to prove that what you've done really has worked. So if you think about this, building this structure was one battlefield. We just let the data compete against itself. But we could also solve this problem in a different way. So rather than including ApoE on the battlefield, we could split it into two battlefields whereby there is ApoE positive data. And again, since it's a risk factor, you have both cases and controls in that data, so you really can still predict disease. and ApoE negative data. And so that would be a way to look at ApoE, because you have the background still is there by the two different battlefields, but you're doing a completely different prediction. So no guarantees, because we're really reducing our power to detect effects, because we're really reducing our sample size. But we thought, maybe we'll get lucky, because this is a raging target. It's almost as strong as ApoE itself. And so it was recovered. When we consider the battlefield that is just people that have no ApoE alleles, that actually ends up bringing up this VGF. It didn't end up bringing up in the VGF in the ApoE positive battlefield, and there's good reasons why, and I'll show that later. So, this is not perfect reproducibility, but this is a way to rethink about data to get, can you find the same target? So what is that target? I'm not going to go deeply into the biology. There's no acronym. VGF is VGF. It means VGF nerve growth factor. It ends up getting to be this highly, highly processed thing where there are a lot of little baby peptides. They're regulating generally in the body and in the brain energy homeostasis, metabolism, and in the brain specifically synaptic plasticity. It was also found by this other huge team called 80AMP. They mapped it. They have not looked at it in the context of ApoE like we have. Because of that mapping, it was also looked at by this imaging consortium. They don't have brain tissue, they have cerebral spinal fluid. So they were able to show that knowing something about VGF could actually help you predict whether somebody with mild forms of cognitive decline were actually going to go to getting Alzheimer's disease. So, yay, we have a new target. We have a new target because we have these structures, but we don't really know what the meaning behind this is. This is just a number. It's just a number of, this is really good for predicting the state of diagnosis, but it doesn't really tell you anything in terms of biology. So this is where the bookkeeping of Bayes comes in. From building all of those relationships, from keeping track of who's winning in the battle, you have something called a conditional probability table. If you take those and take that data for your top targets, and people don't do this, but we decided to do this to get at the why, you can build something that we're calling a conditional probability tree. It's very simple. All it is looking at, from diagnosis to multiplicity of effects, where do people fall? And so the first column here is the probability of somebody getting disease generally in the series. So it should be about 50% because that's the way the cohort was designed. And then you add on whether people have alleles or don't have ApoE alleles. So of course, with the risk factor, it's a risk factor. There should be a higher probability that somebody has Alzheimer's disease in this bin. There's still a probability here with no ApoE alleles because it is a risk factor. But then the glory of this is you can build onto this any new target, and that allows you to check all the relationships. So VGF is scored as low expressing, middle expressing, and high expressing. And why we were really able to recover it in the ApoE negative set is it really makes sense. There's sort of no competition between ApoE and VGF. And it's, you have decreased risk because you have no ApoE alleles, but also you're a rock star because VGF high expression is really happy sauce for your brain. And so these people, there's really a low probability of getting Alzheimer's disease here. Hopefully you can see that. It's 0.08. And then it increases when you get very low levels of VGF. The pattern is a little more complicated when you consider the data from people that have the risk allele. And so this is probably why we weren't able to see it initially, because it's sort of what happens is there's sort of competition between VGF, between ApoE, a risk factor, and VGF, a protective factor. And so when you have ApoE and no VGF, you're really in trouble. But as you start increasing the level of VGF in your brain, you are really helping yourself out. So this is the cell of this, to go back to selling data. What this means is that this target, even if you're in trouble, even if you have risk for developing Alzheimer's disease, if you have the happy juice at a high level, you have a reduced probability of getting Alzheimer's disease. So this is like a very nice target. So I will close there. The conclusions. if you want to be in academic science, you can, it's strife and it's trouble, but you can get there if you are obsessive about your ideas. And I would argue that if you're not obsessive about your ideas, you know, potentially consider you can do a lot of different jobs. But if your love, bread and butter is your ideas and executing ideas, academia is for you. Get good people in your life. There's a lot of toxicity at every single level, but it is super fun, a great job if you run into people that will support your ideas, you know, get you through your failures, and also work with you and listen to you as opposed to shutting you down. That is really the key point here. And also get your set of fools that can make you laugh. That's key. And finally, science. VGF is protective on an APOE background, a lot of more work to be done, but the math really held and it's pretty exciting stuff. These are the people that were involved. City of Hope, mainly, this comp folks, I already showed them. All of the brain banks, you know, these guys, the initial phase 1, they really had to take a leap off a cliff for me. These are all the phase 2, and mainly, you know, people that donate their tissues to science are some weird level of altruism. So I am deeply indebted to the patients and their families. And I will stop talking. I think we have time for a few questions. I don't know if anybody wants to ask questions.
We do. Thank you so much for the fantastic talk. I personally really appreciate the human side of things, because that's the whole goal of this series of lectures, to show the human side of researchers, data scientists. And I think that you absolutely nailed it. And the advice that you gave is so universal. It applies to so many fields.
Because I feel like it's my opinion, but I also feel like I've had a little bit of a weird career. So, what has kept me in this?
It matches almost 100% the advice that I give my students that are about to launch careers in the professional world. Like, just avoid toxic people. Try to find people you match with. I mean, that's so important. Try to support others. I mean, don't be just one seeking advice, but also give advice to other people and help other people as well. Create a community of help, right? That's also super, super, super important.
Yeah, there's actually a guy that studies this. He's got TED Talks, Adam Grant, and he studies how, I don't know if you know him, he studies how people work in systems. Oh, he's great. And he actually says, okay, fine, maybe the competitive narcissism, crazy solipsistic viewpoint, maybe that's a winner. Yeah. And so he treats it as a data problem. And what he has found is something called, it's a lot of different things. There's a lot of different personalities make systems go. But one thing that really struck me is something called the 5-minute favor. So you can't, do everybody else's work for them. But if you can do something very easy for somebody and help them move along in like 5 minutes or less, that is a relationship, that is a connection that you've made. That person, if they're a good person, they're going to go, oh, do you need help? It sort of snowballs. So being good and listening as well, a lot of my ideas come from listening to other people. And I think that is getting lost in hyper competition, and just being decent to each other is also getting lost. I don't know why. It's really depressing.
Yeah, it's absolutely depressing. Make a personal policy not to work with bad people, but only with people whom you love.
I sent one of my collaborators, because they're trying to sort of figure out how to run a department, and I was like, you isolate bad people as if it were a virus. You do it subtly,
that's harsh.
Just send them to Siberia.
Wow.
Some of these people, man, I'm telling you.
Yeah,
I had a guy that come at me that wanted to be both the last author as well as the last and corresponding author as well as the first author. And that was actually good for me because it made me laugh. And I was like, I don't know actually how to type that and the words to put it in a paper.
Anyway, I don't want to monopolize the conversation.
That
was completely my fault, because I really care about these things. Anyway, so the conversation is open. I mean, we are a relatively small group. Yeah. So if anyone wants to unmute yourselves and ask any question, we still have time until the end of the session today. So please go ahead. And if you can turn your camera on so we can all see you.
Yeah, and we can do this as like an AMA, doesn't have to be a science question. It could be for any students in the room, it could be a career question. I am open. That's the whole point of this. So, anybody?
Yeah, I can. Oh, Janina, were you going to say something? Okay. So thanks, Amanda. That was terrific. And like Alberto said, both from the good to see you here, both from the the human perspective as well as the science perspective. And I was just wondering, so many of us have experienced the kinds of dastardly behaviors that we're talking about, right? And it's interesting that so many people can relate to it. And as Kissinger said, the politics in academia is the most vicious because the stakes are so low, in a sense. And I mean, you just see that over and over again. So my thinking is, why don't those of us who feel this way come together and actually stand up to the crap?
I think, there are problems. So why Adam Grant's model is a 5-minute favor is that a 5-minute favor, 5 minutes is not going to wait on you. So I think the problem is the hyper competition for grant funding, and that in order to get grant funding, you have to be very solipsistic and me, I'm the best, my science is the best, you got to sell yourself. And so if you dive too far into somebody else's stuff, you can lose your own path. And so I think peer review, I think hyper competition has set up this model. But I would argue, I agree with you, we should fight back. But it's sort of this balance of, I don't want to lose my own research because I am so invested and I found cool things. Why wouldn't I follow it? But if somebody needs help that I can give them and it's low stakes for me, then I think we should always say yes. Again, with the, it's limited time. Again, you have to really be like, I'm the best and my stuff is the best, but helping people out, if it's not going to drag you down, I think is a really great thing to do. And I try to do that. I write people, letters of recommendation. I talk to students, every single bit of keeping the eyes on my own prize, but also if it's low stakes, helping other people out. I think that's actually helped me. It's only done positive things for me because it makes that network of people. So, I agree with you with the caveat of don't get lost in helping other people that you can't do your own stuff.
And also, I think about to do it compassionately so that we get together, spend a little bit more time than 5 minutes, because it's easy to say 5 minutes, but sometimes for things you really care about or you really believe is going to help a lot of people, God damn it, you go ahead and put in more than 5 minutes. So that's what I would say. And secondly, to do it compassionately so that you're not trying to bring anybody down and sort of just perpetuate the nonsense, but to compassionately invoke that humanity that lies latent in most people. In some people, it may just not be there, and that's fine. Those are the ones you send to Siberia, like you said. But others, and I just don't understand why we don't do it, because it's horrible, what's sort of going on in the name of academia. I teach, mind-body medicine, amongst other things here. I work on stress, on good stress as well as bad stress. And I see the completely unnecessary devastation that this job-related stress in academia and outside creates. And to me, it's absurd. I mean, it's ridiculous. And I just wish there was some way for some of us to come together, form a nucleus of people that sort of compassionately begins to say to the folks who are imposing stressors on others that, hey, let's just back off. At the end of the day, if you're struck with something really, really bad, you'll quickly realize that all this stuff you were doing to people was terrible, and completely unnecessary.
I would actually like to bring Nick in on this conversation, our fearless leader, because I do think part of the other thing that breaks down is leadership. Leaders have to model the behavior that they want to see. So if they are being mean, other people are going to be mean. And I have to say, again, one of the things that I really appreciate about the IDSC is, as much as they can, they really try to help support people and weird people like me that don't necessarily fit into data science. So with that little bit of, thank you, Nick. How do you feel about the problem?
You try to circle me in, I think, Amanda.
Yes,
right?
exactly.
But this is your show, not mine, right?
But how would you lead? How would you lead in a way that fosters this kind of thing.
What you are saying is so true, right? So, and in some cases, even we select our leaders actually because they have gone through this hyper competition type of thing, rather than being what I would consider a good leader is to build an organization and build the people and making the appropriate, how can I say, resources, investments, and so forth, so the other people can be successful, not the leader. So that's how I would describe this. And what we're trying to do actually in iDisk is, as you know, Amanda, is pretty similar kind of thing, right? We try to build an environment where people can interact, can be themselves, can find things, and in my opinion is that the reason that I got into science is not necessarily because of some kind of credit. We all understand the business of science, that we need to get grants and so forth, but we all got into the science because we feel passionate about it. And we want to be surrounded by people that they share the same passion. And a little bit of competition is not necessarily a bad thing, but it's meaning, in terms of, how can I say, hey, look at what we found. But the question there is that what you are saying here is, it's very important, is that how one builds an organization in an academic environment where you are actually bringing in the best of the academia and not the worst of the academia. So what we're trying to do in iDisk is actually to bring the best of academia and the best of, if you will, the business or industry aspects of how we can all go together and solve a problem. Now, what it happens in many cases is that actually instead of bringing the best together, we're bringing the worst together. And this is where things go really very bad. And we're trying to avoid
by best, because I think best is a combination of people that are excellent at their field. But so, for example, it's funny that you say isolate people to Siberia. My colleagues at City of Hope, again, this is one of my favorite collaborations right now. The lead guy, Andre, is from Siberia. But the reason why this works is, you could look at them and say, well, they're not in the 80 amp, they're not super sexy or whatever. But why it works is that they're actually super rigorous minds, really heavy thinkers, not, like popular or whatever that is. But they're willing to question and work on problems and keep going together. And, conflict resolution is like, we don't need to have a people's court in this collaboration at all. And I've been in collaborations where I felt like we should just go to people's court and fight this out because it's so toxic. So I do think that there is, in terms of thinking about good scientists, there is who is it that just left? Lander was just fired, and Eric Lander is a brilliant mind, but he couldn't manage a team. And so I think there is this aspect that we're forgetting.
That was a very well-known fact for decades.
Exactly. And, that doesn't denigrate his mind and his consciousness. Eric Lander, for people in the room, is this guy that has done a lot. He's came at it from a math aspect, but done a lot in the genetics field. Very sort of famous and smart and everything. And he was sort of put on high and then summarily fired because he was really toxic with his team. And so it actually was a female lawyer that pointed this out. And she had the guts to say, this is not going to work. And so he got fired. And, he's H index of whatever, papers, citations, everything. But it seems like, and I think this is really good, that, we might be addressing this problem of there is, the sexy superstar, but if the sexy superstar can't work with people, then it is a question as to whether we're getting the best product out of the day, because part of science to me is communication, is thinking about problems together. People asking dumb questions is almost exciting in a way, because then you're like, oh, okay, so I explain it this way, and then they ask something else, and then you just move on and move on and move on, and that sometimes generates ideas. So I think people that are patient should be valued, people that are, kind should be valued, and, not just when you say excellent people, I get you, we need to have superstars here, but superstars in the context of there are great minds that are also decent. And I feel like sometimes that plot gets lost, but
That's
How
an
refreshing.
excellent, the point about decency, I think that it's absolutely essential.
It's weird. I mean, I don't know if it's because my dad's Canadian and I come from that background and everybody's apologizing and saying sorry, but I have been in spaces where I'm like, what is going on? I don't recognize this. I think it's stress. Fergus, write your next grant on this. Why do people behave badly when they get so stressed out?
Well, so I was just teaching residents yesterday morning, and I showed them this snippet from Star Wars: The Phantom Menace, where Yoda, when deciding whether little Anakin Skywalker should be trained to be a Jedi or not, says that fear is the path to the dark side. Fear leads to anger, anger leads to hate, and hate leads to suffering. And so this whole cascade, not to excuse bad behavior, because many people are fearful and angry and they don't go making life hell for others. But this cascade of stress, anger, fear can sort of become this deleterious feed forward loop. And some psychosocial situations, such as ours in academia, are particularly conducive to doing that. But they can be, and anywhere where you nip those deleterious feed forward cycles, you can kill the cascade. So it gives you actionable targets, although it's easier said than done. So I believe very strongly in not just talking about these things, but really having good people come together and gently, compassionately trying to sort of change that system. And in some cases, it may not be as intractable as it seems.
Yeah. I mean, I think again, it requires strong leadership and the space to have some quiet time. Again, I would not have had my career if I hadn't been sipping. John said, make me lattes because I'm too busy to make my own latte. And I said, I will only do that if I get time in the morning with you. And so we had coffee, we talked, we trust each other. It ended up starting a whole field of science. And so the quiet spaces, which I don't think protected time is so important, and it's really hard to, get institutions to realize how important protected time is. But it is what it is. The systems are the systems. We will continue to do our best. But I don't know if we want to open the floor to anything else. Anybody in the...
It's almost out of time. If there's any other questions, feel free to.
I have a quick one, data-related one.
Sure.
May I?
What's up?
Okay, so we have, and maybe this is a problem that can't be addressed. So we have really nice data, but I don't really have any tools to analyze it with. So I'm always dependent on other people. But it's often a question of small numbers, large numbers of variables, yet with prior knowledge to guide the analyses. Is there some way that folks here who can help and ideas to tackle that?
I don't know. And I'm not really good at that problem because I have, lots and lots and lots of samples, and that's how we address the problem. But there might be, maybe that's a problem for machine learning. I don't know if, Nick wants to jump in on this, or, Tim, you as a programmer want to jump in on this. I think this is true of the university as a whole. Is there are a lot of people that are running around trying to figure how to data. They're new to big data and they're trying to figure out how to manage big data. And, a lot of people are asking me this question and I'm like, I can't do it all. So take the lead. Take the lead, Nick.
Sure. Again, I mean, we'll be happy. I mean, there's a number of different people here actually that they can potentially provide advice and see what it is. So, I mean, I can pick people from here that I see here that they can, but there's a number of different people that they can definitely look. But also I have to admit though, and I have to say that the devil is in the details, right? So one has to, I mean, this is the issue with the data, right? So you really need to understand. And sometimes you have a lot of data that it's really non-informative. And sometimes the data is not as much, but they can create a lot of information. So you need to understand a little bit about what you have, what you do, and how you can do some of these things. So this is a very broad thing to say. Yes, there is a number of different folks here that can help with that. Melvin, I can pick Melvin here. Who actually wrote it on the chat, and you can see, for example. right? And
Melvin, would you be willing
Yes.
to? Here's my 5-minute favor.
Yes, actually,
today.
I have a little bit of a problem with my voice today, but I'm going to try to explain what we actually do when we have a lot of variables. We do a feature analysis and reduction. We try to correlate the variables because most of the time we end up with a lot of columns in the spreadsheet, which we don't need. So actually, there's a lot of correlation sometimes, and there are methods to deal with that. So to deal with the immensity of columns, and by doing correlation, we can just reduce the data set and remove columns that we really don't want. That is called feature reduction.
right. So this is what Melvin was talking about this, and this is why I'm saying that perhaps we should talk about this a little bit more and see the data and so forth. And again, I don't want to take time. You know, we are a few minutes past, but I think we can talk. There is a feature reduction. There is, machine learning aspects in order to extract the features. There is all sort of different techniques, but also depends on the data as well, right? So that's, but Melvin is absolutely right. So one of the ways that we try to do is always, and actually Amanda has been talking about this a little bit on the periphery, is that how you do feature reduction, right? So, how you can actually find the important components that, and you can separate from those.
Yeah, okay. Melvin, would you be willing to, I don't know,
Yes,
this is not my
I
purview,
will.
but yeah, okay, perfect. Okay, there we go. Made a connection.
Mm-hmm.
5-minute favor done.
Well,
and this is the kind of thing that needs to happen here.
Well, thank you so much
Of
again,
course, of
Amanda,
course.
for making the time. I think that this was great. Both your learning about your career, learning about your thoughts about what a successful career entails and what depends on. I think that it was highly informative. I would just also like to thank everyone for being here for another one of our data science series talks. And now we will all see you in the next one.
Thanks, everybody. I really appreciate
you all.
your time.
Thank you.
Thank
Take care.
Bye.
Thanks.